Hello, my name is Anthony Maresso, and I'm an associate professor at Baylor College of Medicine. What I'd like to talk to you about today is related to the current situation that we're in with this viral pandemic, but has to do with another microorganism, that is bacteria. And it is a pandemic that has been ongoing for quite some time and will continue to be ongoing well into the future. It's a more slow pandemic, one that is gradually gaining steam, but is of no less concern and potentially could be more impactful as we move forward into the next few decades. So the title of the presentation is Chasing Change, because what I'm going to try to describe in this series here is that the organism in question has the ability to evolve very quickly, and that undermines the drugs and prophylactics that we throw at it. So a moving pandemic and an adaptable medicine as potentially a solution. Bacteria have always been with us. They are some of the original organisms on this planet and are very, very successful. So successful, in fact, that they have maintained themselves in their current state for several billion years. But as eukaryotes evolved, and human beings too, bacteria learned to, in a way, symbiotically become affixed to human beings, but also in some contexts cause disease. And so this is a picture that shows, I think this is leprosy, meant to represent these boils, but really is applicable to any bacterial plague. And we've had several throughout our history, the most famous one being in the 14th century, where it's estimated a third of the population of Europe was killed. And up until recently, we really had no real way to deal with bacterial infections. They were a scourge of our species, one of the leading causes of death, along with viral infections for a long time, until the last, I think, about 60, 70 years, where cancer and cardiovascular disease have now come on top with infectious diseases, worldwide mortality about a third, I believe. But it's still substantial. Tens of millions a year, people are killed by infections. And so this picture kind of represents the state of these plagues when we had no ability to really deal with it. Everybody pretty much knows that antibiotics are life-saving medicine, and this is an old picture of a pharmacy putting up a advertisement for penicillin, the wonder drug that can kill bacteria. And it issued in an era shortly after its discovery of biomanufacturing in the 1950s, chemically manufacturing in the 1950s, in the late 40s, that brought in the golden era of antibiotics, where we essentially made 4, 5, 6, 7 different classes of different antibiotics, all different permutations of it. And these antibiotics were life-saving medicines. And they drove the pharmaceutical industry through the 60s and 70s and 80s, and sort of opened the door for other drugs, for other diseases to be pipelined in such an efficient manner. And so antibiotics are one of the ways in which we deal with bacterial infections, something that I call the Trinity of antibacterial technology, which is Essentially, civil engineering is the first line of defense, where your water or your food in the past and throughout most of our history was contaminated with life-taking microorganisms, but now is quite clean. And for most of the world, you don't really have to worry about it so much, but there are still developing nations where this is still a substantial concern. Then there are vaccines. That's the second arm, and or the second part of the Trinity. And we have good vaccines for like tetanus and pertussis and diphtheria and botulism. But we don't have vaccines for some other bacteria. We'll talk about that in a second. But these two alone have put a major dent in the number of bacterial infections that people will die from. And then antibiotics came along, and that was sort of the third part, that all three of these together really form a great defense mechanism, societal defense mechanism against bacterial infections. This is just an example of the power of antibiotics right here, where this is an antibiotic put down. You can see this zone of clearance. This is a bacterial lawn, so bacteria growing around this, and they're just killed. It's just wonderfully potent, amazing drugs that work so well. But we have a little bit of a problem, and this problem is, I think, underappreciated because it flies in the face of the old paradigm. The old paradigm was that you might acquire a bacterial infection from an outside source, external to you. Think of getting cholera, for example, when you drink water, contaminated water, or some other outside-in entry mechanism. But this picture here is actually from my laboratory. This is in what we call an intestinal enteroid. We take these stem cells from patient biopsies, and we grow these sort of mini organs, mini intestines in the laboratory. And you can add bacteria to them, and bacteria that live in and on us, they really bind to these things very well. And you can see that here. This is E. coli, and this part's U. And they form these biofilms, and essentially they will colonize you. And this is true throughout your body, not just your gastrointestinal tract. There are billions or maybe more bacteria in your gastrointestinal tract, but they're also on your skin, and they're in your nose, and they're in the urogenital system, your eyes, your ears. And so this is called the microbiome, and it's recently been appreciated as a essentially ecosystem of contaminating it in a way, bacteria that live on you, and they come along for the ride. The problem with this, historically, the microbiome wasn't really much of an issue, but you see, we're living much longer now, right? The life expectancy is from 40-something years, 30-something years, not sure, a couple hundred years ago, to pushing 70, 80, 90 in some parts of the world, and much past what was naturally possible. So it's giving a chance for these bacteria that would normally not cause a problem to now be a problem. Because the miracles that are keeping us alive, the medicines, for example, sometimes they make one immunocompromised, or just because you're have so many things that have happened with you, or you've grown sort of older, become more susceptible. And so the paradigm of the future is one in which these bacteria are going to, that live in and on us, are going to be the major drivers of substantial life-taking bacterial infections. And so there's this disease called sepsis, which it doesn't get a lot of attention, but in the United States alone, every year there's about 1 million cases of this. And depends on the year and how people record this, but there's about a quarter of these that don't end up well. Unfortunately, the patients die. And it's where these bacteria that live in and on you, they kind of go crazy. They grow, they use you as an incubator, they'll spread throughout your body, mostly in your blood. And in some cases, the immune system tries to deal with this and uh, puts it into overdrive, and the collateral damage is large scale tissue destruction, including major organs. So the bacteria plus the immune response is thought to sort of take your life. Some of the bacteria that do this are listed here. E. coli, Staphylococcus, Enterococcus, Pseudomonas, Klebsiella. These, you don't really get these from social distancing right now is a sort of popular thing to do to sort of avoid this coronavirus. And we can't really social distance these bacteria away. You acquire them early in life and they stay with you. You might acquire some bad strains later in life, but these things sort of make you their own home. This is a challenging thing to do. But what's the real problem? The real problem is as I mentioned, this new paradigm. People are getting older and being kept alive longer than they naturally would. But the remarkable thing about microorganisms is their ability to change in real time. And to not only change for their own benefit in whatever little system they find, but their ability to change in response to the measures we throw against them. And so I'm just going to talk to you really quickly about something I like to call the mutagenic Tetras effect. And I bring up an example here that in your gastrointestinal microbiome, it's thought that every day, just because of the normal process of replication, you generate 9 million new mutant bacteria. Now, these are not new strains. Technically, one single change in the entire genome can be called a mutant, and not all of them are the same mutation. Many are different, but some are redundant. But it's estimated there's about 9 million. So just take that with a grain of salt. Some caveats there. If half of that is true, and this occurs every day, that's about 1.5 billion mutants per year. Well, let's say there are 7 billion people on the planet. There's more than that. That's about 10 to the 18th mutations per year. Now, granted, they're not all the same, but even if it was 10 to the 15th or 14th or 13th, whatever, these numbers are astronomical. It doesn't matter, right? You only need one single mutation to become resistant to a chemical antibiotic. Just one. That's well established scientifically. We can mutate certain enzymes that bacteria make, and that enzyme will now acquire the ability to destroy an antibiotic. So this is a math game that we are losing. And we're losing it because bacteria have this great ability to do it, but also because the drugs we make don't keep up. So this is the mutagenic Tetras effect, right? Bacteria in particular will mutate in 4 ways. So there's de novo mutation, which is just what I sort of modeled here, where you just acquire a SNP in the genome, a single one change in a base, and that will confer a new property to the organism. But that's not the only way they change. Bacteria acquire whole genes that will destroy antibiotics when they make the proteins of those genes, destroy antibiotics from their friends, other bacteria. That's called transformation. Usually that's taken up as a plasmid, but they'll also do this in a form of primitive sex called bacterial conjugation, where information will be transferred from one cell to a donor cell. And they can be infected too by viruses and going to get into that in a second because that's where I'm leading. And these infections will also deliver new genetic elements. So 4 interacting parts here. Tetra, sect, that intersect, and that are mutagenic. This is a concept to think about that's really important. It is lost on those that I think try to develop new antibacterials. So here's an example of E. coli that really rapidly destroyed or got around the Trinity of antibacterials just by evolution and in a short period of time. So I call this the Tetraceptin Carnate. Okay. Sometime late 90s, acquired a new adhesin, acquired some transporters. This is probably through conjugation or plasma-mediated uptake of genes. Horizontal gene transfer, we call that, from one to another. And the acquisition of advanced ability to adhere to your gastrointestinal tract where E. coli lives and to take in nutrients essentially means it's going to be able to stay with you to a greater degree. It's adapted to you. It's going to less likely to not colonize, more likely to colonize. And so that kind of eliminates civil engineering because once you get it, regardless of how you got it, it's going to stay in your GI tract. Right? It's colonize you permanently, potentially for life. And then this E. coli, for reasons I'm going to get into, acquired resistance to antibiotics. And so that kind of makes the antibiotics then useless. And because E. coli is E. coli, it likes to take parts of its genome and give it to others and take in parts of other genomes. Acquires many, not just one or two, which is what we typically make, how we develop vaccines against one or two toxins or targets, and sort of take that arm ability away from the organism, and now it can't function well. Well, when it has many of these so-called virulence factors or toxins, it's hard to develop a vaccine. There's too many targets to go after. And so it just consumed, spit up and chewed out your trinity of antibacterial therapies. So why is this a problem? Well, with antibiotics in particular, because we use so many of them, so many antibiotics. Essentially, I think my calculation here is 380,000 people's worth in weight antibiotics a year. And that's enough to fill up 10 stadiums, average size baseball stadiums, just to give you an idea how much antibiotics we use. This may be in America alone, I'm not sure, but you get the point. We use a lot. Half of that's roughly distributed between people that need to get antibiotics because we think they have a bacterial infection, they're prescribed it. And roughly the other half goes to agriculture. We used to put antibiotics in toys and all types of things and kind of learned not to do that anymore. So ag and people get most of these drugs. So it's all over now. That's good in a sense because we've learned to control bacteria with them. But again, the mutagenic tetrasect comes back because Darwin's great theory of natural selection, right? If you create a pressure that an organism has to adapt to, only the ones that are best adapted for that pressure will survive, and those that are not will perish. So here's a petri dish, just as an example. You have green bacteria and you have gold or yellow bacteria. You place an antibiotic stress on them, and let's say it's an antibiotic that kills the gold bacteria, the green ones survive, right? And so what are they going to do? They're going to occupy the niche. They're going to expand and fill in where the gold ones were. They're going to grow in the presence of this new selective mechanism they're more fit to. And so they dominate, they come to dominate the situation. And this is what they are doing to us. They are dominating our microbiome. They're growing and increasing in number as a part of our microbiome. And so we're setting us up for a real problem. We know that these drugs are failing year in and year out, and we're losing the ability to use many of these drugs to control these bacteria for real bad infections. And so either we change the way we develop and approve new drugs, or we develop and approve new drugs that change. And these aren't necessarily mutually exclusive principles, but they are new ideas. The current pipeline, the way we approve drugs, takes a long time. And so I'm going to get into that in a second here, but I want to introduce to you something called phage, which is a virus. It's a virus that infects bacteria. It's like the flu for bacteria, which is amazing when you think about it, that the plague itself can have a plague, but that's the beauty, of course, of life on Earth and evolution. Takes advantage of these things. And so phages are something that have been around for forever, billions of years, but they were only recently discovered in the early 1900s. And this virus, when you mix it with bacteria, they just blow these bacteria up. They infect, and they make more viruses, just like what viruses do. This is what SARS is doing to us right now, right? Infects us, make more of itself, and then spread these viruses. These phage do the same thing for bacteria. And they kill the bacteria in the process. And so early on, it was thought we can use these phage as drugs, and we were starting to for bacterial infections in people. But then antibiotics were discovered. That industry blossomed and grew and became a wonderful industry. It was cheaper and easier to make antibiotics. And so phage went in so-called silent years where we didn't think about them anymore. But now that antibiotics are failing, now that resistance is growing, we're in this resurgent session where we're again turning to whether or not phage are able to potentially be used as drugs. And they are. Every day now around the world, phage are given to people, but they're given to people in a very limited sense because we're still learning about whether or not they can be really good drugs. But I want to introduce to you why we're excited about phage and why this paradigm about drugs or the pipeline changing, I think, is valid. And so on the left here is penicillin, very fixed chemical structure. It cannot change in real time. Cannot change in real time. It's fixed. This is a very potent drug, but it's starting to fail. It has failed for a long time. When it fails, we can add chemical constituents to this to improve it, and it will work again, but then the bacteria will evolve around that. And then it takes a long time to make new drugs. So we can't put so many changes on these. We get limited in how many we can do, and costs a lot of money. This is a lower end, 250 million to just develop one drug. It's probably more closer to $1 billion now, and maybe 10 years on average. Well, these bacteria, we already told you, they evolve in days, on a time scale of days with a selective pressure. It takes 10 years to develop a new drug. And that's because we're trying to fit these things to chemistry, fixed chemical structure. Phage are different. The basis of their change is not a fixed chemical structure. It's nucleic acid, it's DNA, it's RNA, it's mutation. So in a way, we can use the same processes that bacteria use against us to get around our drugs, mutation and diversity, and turn the tables right back on bacteria. Because phage, the basis of their change, is mutation too. And so you could say you have a phage and maybe it's not working against bacteria. Bacteria become resistant to it, and you could turn here this green mutation to a red, make a new phage, and now it infects that bacteria. And quickly, very quickly, on the order of hours, we've learned in the laboratory. Or find phage that already have this mutation in the environment and can be brought quickly to address the new resistance strain. And you can calculate the number of permutations, the number of possibilities with a certain sized genome, and the number of mutations that can occur, or with the diversity of the number of phage in the bacteria, essentially infinite, almost an infinite number of resources of bacteria that we can find. So there's precedence for the adaptable drug to come. It's thought that you acquire through one's lifetime 10 million mutations in your own cells as they divide per year. And that, of course, can be very, very bad. It can lead to a very well-known disease, cancer. And so, this is sort of evolutionary mutation in real time. But we're using adaptable drugs, drugs that we can change. It's CAR T cell therapy to address cancer. Right? So this is an example of a medicine that changes. It's not the only one. Everybody's heard of CRISPR and gene editing. Well, they're developing CRISPR constructs that fix specific mutations in one's genome. That too is a very personalized or adaptable medicine. And you can change the construct to adapt to the mutation that is in question. And at some point, I have no doubt that we will, as organs fail and things like that, we will adapt one's own organs to replace their own. Right? And that'll be a very personalized or adaptable medicine too. So why not for infectious diseases? The disease that is most adaptable, these viruses and bacteria, why don't our medicines adapt to address them as well? And so that's the take-home message from me. So some key targets: infectious diseases are a moving target. They will always be. Bacteria and viruses mutate often. Some of these mutants will have a fitness advantage and they will propagate and get around our drugs. So our medicine should be equally adaptable as well. That's the vision moving forward. That's the paradigm we wish to address. So phage may possess diversity and evolvability to meet this need specifically for bacteria. And the principle can be applied to other diseases, right? And we're already sort of learning that for cancer. So evolution is to summarize, this most powerful director of change. It's given us the diversity of life we see today, and bacteria and viruses have mastered that. We may have to change our thinking as they change to our measures so that we can adapt faster, right? And that might be the paradigm moving forward. And there's got to be a lot of science that has to be done to get at that. This is not something that we can just walk into lightly. We have to really learn a lot about this, but it's one that I think we should invest in the future. So I hope you enjoyed this concept presentation, and we'll be doing more of these and putting them out there on our social media channels. Take care.